In its simplest form, sunburn is the overexposure to UV radiation from the sun, resulting in radiation burns of varying severity. This radiation causes DNA damage and subsequently type 1 cell death, meaning that the tissue is destroyed and replaced. Two types of UV light can cause this damage, UVA and UVB. UVA has a longer wavelength, is responsible for aging, and can penetrate windows and cloud cover, while UVB has a shorter wavelength, it more readily causes skin burns but with weak penetration. Both types can cause DNA damage and skin cancer as a result. The damage caused by UVA and B is usually through the formation of a thymine dimer, which is the kind of molecular lesion caused by light or photolesion. Lesions are damage to the structure of biological molecules such as DNA, causing a reduction or absence of a particular function. Incoming UV photons from the sun cause covalent bonds to form between consecutive nucleotides on the strand of DNA, meaning they bind together and the typical base pairing structure of the area is destroyed. This means that the sequence of amino acids encoded by the DNA changes and a different protein is produced, otherwise known as a mutation. When this damage occurs, four things are usually triggered by the body as a defense mechanism. The first of these is DNA repair. The body does this through repair enzymes called photolyase enzymes, which detect lesions and catalyze the process of photoreactivation. In photoreactivation, photochemical reactions occur to remove and restore the damaged sections of DNA, using light of a longer wavelength. The body has other repair mechanisms, such as nucleotide excision repair, in which the dimer is removed and new DNA is synthesized to replace that in and around the section of DNA affected. However, if these dimers are not repaired, they may lead to the formation of skin cancers such as melanoma, basal cell carcinoma, and squamous cell carcinoma, all of which are predominantly caused by sunburn. If DNA repair is unsuccessful, then apoptosis will be triggered. Apoptosis is a type of programmed cell death, meaning that events inside the cell trigger it. Necrosis, on the other hand, is unprogrammed as a result of external factors like trauma or infection. Autophagy and necroptosis are also forms of programmed cell death. Autophagy occurs within the cell and is used to remove and recycle dysfunctional cellular components, and is highly regulated and controlled, as is apoptosis. Necroptosis is thought to be a programmed form of necrosis, to be used as an alternative to apoptosis if apoptosis signaling is interrupted by pathogens or mutations. During apoptosis, the first step is that the nucleus of the cell shrinks. Then, blebbing occurs, in which parts of the cell within the plasma membrane bulge out and protrude on the surface. After this, the nuclear DNA is broken down, and the organelles break down and form fragments, which, once the cell breaks apart, are enclosed in sections of membrane. Finally, immune cells collect and remove these packets, thus preventing the contents from spilling out and causing inflammation, as occurs during necrosis. In apoptosis, there are two ways cells can be signaled for deletion. The first is the intrinsic pathway, which is cell suicide due to cell stress, from extreme temperatures, chemical conditions, toxins, or mechanical damage. Then, there is the extrinsic pathway, which is cell suicide due to external signaling from immune cells. These pathways pull the trigger by activating a group of proteases called caspases, of which there are initiator and executioner caspases. Cancerous cells which eventually form tumours manage to evade both these pathways, allowing them to survive and divide, accumulating mutations as they go. There are four primary reasons why apoptosis is necessary. The first is during early development, where cells need to be removed to form specific structures, for example, to separate fingers. Also, cells with irreparable DNA damage are eliminated to prevent infected or cancerous cells from dividing further. The third function is to maintain balance. Just as some cells are created, others need to be destroyed, especially those which only perform temporary tasks. Another purpose of apoptosis is for immune function. This is because new B and T cells are tested to see whether they will react to the body's own components and are destroyed if they do so, thus preventing autoimmune conditions. Maintaining a healthy balance of apoptosis is essential to our health, as too much causes atrophy, where areas of the body waste away, whereas too little causes cancer. The third, and probably the most obvious defense mechanism to combat damage, is peeling. When our skin peels, irreparably damaged skin cells in the epidermis are physically removed from the healthier tissue underneath. The final defense mechanism against radiation burns is an increased production of the pigment melanin. Melanin is a natural skin pigment which acts as a photoprotectant, meaning it absorbs UV light and intercepts the photons which can cause direct and indirect DNA damage. Indirect DNA damage occurs when the UV photon excites the chromophores on the skin which originally absorb it. This induces reactions between the excited chromophore and DNA, or the chromophore and other molecules to form free radicals, which may also diffuse and reach the DNA. Chemicals which remain in this excited state for a long lifetime may travel in the body and cause damage to other areas as well. There are 3 types of melanin, which correspond to different coloured pigmentation in the skin. The first type is eumelanin, of which there is brown and black eumelanin. Pheomelanin is found in the pinkish parts of the body, such as the nipples, and red hair is a result of equal parts eumelanin and pheomelanin. The final type of melanin is neuromelanin, which controls the colour of neurons and is found in the brain. People with red hair contain lower levels of melanin, meaning that they burn more quickly, tan more difficultly, and absorb more UV, meaning less exposure is required to synthesize vitamin D. When people tan, they produce either brown or black melanin, causing the skin to darken and resulting in an increased protection to the sun of around SPF 3. However, this doesn't exclude the possibility of DNA damage, as no tan is safe. This sun protection factor can be increased through the application of products like sunscreens, which, similar to melanin, work by absorbing or reflecting UV radiation. In terms of the symptoms of sunburn, some are more obvious, while others cause more internal damage. Firstly, you may notice inflammation triggered by the DNA damage and subsequent cell death. Erythema, or skin reddening, is caused by the increased blood flow to superficial capillaries as part of an immune response. Other symptoms include an increased sensitivity to heat in the affected area, as the activation threshold for the heat receptor TRPV1 is reduced from 43 to 29 degrees Celsius. There is also increased sensitivity to pain in the affected area, as the protein CXCL5, which activates nerve fibers, is overproduced. In extreme cases, edema or swelling may occur, where fluid builds up in the tissue and results in the skin feeling tight. In severe cases, there may be blistering and feelings of fatigue, nausea, dizziness, fever, or itching. Next, let's talk about freckles. These are clusters of melaninized cells most frequently seen on people whose skin falls into 1 or 2 on the Fitzpatrick scale, indicating a fair complexion. Freckles are caused by sun exposure or genetic factors, which may make some people more susceptible to them. In freckle formation, organelles called melanosomes contained in the melanocytes and responsible for the synthesis, storage, and transport of the pigment melanin are transferred to the keratinocytes, which make up 90% of the epidermis. Freckles are often more prominent in the summer and can fade during the winter as well as with age. While freckles are harmless, they may resemble skin cancer, so it is essential to know when to see a doctor and the specific warning signs on the skin. Luckily, there is a simple process to follow to determine whether a spot may be harmful, called the Abcde Method. A stands for asymmetry, referring to how even in shape the spot is, and B for border, as cancerous spots usually display multiple irregularities. They may also be a peculiar colour and have a large diameter more extensive than that of a natural freckle. The final letter, E, stands for evolves, as if a spot changes with time and the letters mentioned become more prominent, it may be a sign of tumour formation. Even if a spot displays just one of these symptoms, it is crucial to get it checked with a GP, as skin cancers are among the most common types of cancer worldwide. If you are not sure, there are many useful resources online, and it is very easy to find out the UV index at a given time and day to adequately protect yourself.